Is this thing on? All right, it's Rutherford here. We're going to teach you about the M&M quizzes, test number 5, mitosis and meiosis. The whole point of these processes is that they are making cells, but it depends on where in your body you're making cells and what cells that you are making, okay? Meiosis is when you are making cells for sexual reproduction that you could pass on to your offspring, which could potentially lead to evolution. Whereas mitosis is when you are just making new cells in your bone marrow, your skin, or when you're an embryo, need to make lots of new cells. And the process of mitosis is described over here with the 5 phases. But let's knock out meiosis first. Meiosis is occurring in your gonads, the ovaries and testicles, in order to make gametes, the egg and the sperm, so that you could be ready for sexual reproduction. The egg and the sperm are special cells. That's why they have to go through this special process of cell division to make the new cells that are haploid. That means they have half a set of DNA. And if they have half a set of DNA, of course, we want the egg to have half a set and the sperm to have half a set so they can meet and fertilize so they can have a full set. That would be called diploid. And the zygote is the goat, the first original, greatest of all time cell that is going to go through lots of mitosis in order to make you as an embryo, fetus, offspring, baby, right? Okay? So the way that we are going to go through meiosis is very similar to mitosis, but we need to understand that it's a slightly different process that is occurring over there in our sex cells, okay? Now, mitosis is a 30-minute process, actually like quite fast. And there are 4 phases, and then there's a kind of a finishing phase. It's not technically part of mitosis, but cytokinesis we will talk about at the end. In mitosis, we have prophase, metaphase, anaphase, telophase, PMAT. In PMAT, prophase, first we are going to pack up the DNA into chromosomes. It was chromatin before. When it is in the nucleus, it is chromatin. And that's when it's just like a normal cell doing its job, making proteins. It's like unbundled. It's like spaghetti inside of the nucleus. That's chromatin. Now, if you're going to move your DNA, which you will be doing here, you are going to pack it up into chromosomes like you do anytime you move anything. Okay, you got to pack it up. Now, you're not going to be able to move it if it's stuck inside the nucleus, so we got to break down the nucleus, and then we're going to build some protein fibers, which will be moving. These are our movers and our moving trucks, which are going to be moving the chromosomes to the middle. Now, the middle is metaphase. You're going to use these protein fibers to line up the chromosomes in the middle of the cell. Now, we line them up in the middle because we're going to split them in half during anaphase. We're moving them apart. So these protein fibers are going to be pulling and ripping these chromosomes apart so that they can be on the left, the right, or, from 2 different sides. And then that's telophase. Telo means end. And so we're going to wrap up the DNA, which has already been separated. That's our whole goal, is to take one cell and divide it into 2. So we've already separated the DNA from anaphase. So telophase is going to wrap up that DNA in 2 separate nuclei. And then you can let the protein fibers get broken down, and you can build new nuclei to keep that stuff safe. We've already moved it, okay? So let's go ahead and wrap it up in nuclei to keep it safe. Then once it's protected in the nucleus, it can go back to chromatin from its chromosomes. And then, of course, you just split the rest of the cell. That's our cytokinesis there. This is the process of just making a new cell. This happens in your skin. It happens in your bone marrow to replace blood when it gets lost. It happens when you're creating scar tissue, when you are going through a growth spurt. This happens when you are an embryo, when you just need to make a lot more cells and you're developing over 9 months in your mother's uterus. Okay, this is a process that just goes on over and over and over. Now, not right now as an adult, you don't go through it everywhere all the time, okay? But it is something that is there for you whenever you need to make a new cell. Now, the first thing you got to do before mitosis to make sure that you are ready to split and have 2 separate cells is DNA replication. Now, that DNA replication takes 8 hours. You're going to take that DNA, unwind, unzip, and then you are going to build DNA on both sides of the exposed. Then you are going to be able to have 2 full sets of DNA. Okay, that's double diploid. That's what's called quadruploid. Now, if you are double diploid and you are quadruploid after DNA replication, that means you can go into mitosis, split them in half, and then you will have a diploid and a diploid cell. And you can do that over and over and over, and each cell is going to have a full set of DNA. Now, the full set of DNA for humans is 23 pairs of chromosomes, 46 total, okay? Now, the reason that DNA replication takes so long, 8 hours, is because there are 3 billion base pairs. So when we draw DNA, we think about the ladders in the double helix. Every step in the ladder is a base pair. The A pairs with T, the C pairs with G, and every step in that twisted ladder is going to be a base pair. There are 3 billion of those. So it takes a long time, 8 hours for us to do that. We must do that because, of course, when we're going to split, we're doing cell division in meiosis and mitosis, we are going to need to make sure there's enough DNA once we go ahead and go through that split. Now, if you can imagine, there might be some mistakes occurring there, right? 3 billion base pairs, these letters, that's where our mutations come from. This is where we get the first human with blonde hair, with red hair, the first human with blue eyes. Their parents don't have it, but there must have been some sort of mutation occurring in their gonads, the ovaries and testicles, when they were doing the DNA replication. If there's a mutation there, then that can lead to an offspring that has some genetic variety different than their parents. And that, if it is beneficial in your specific environment, is called an adaptation. Therefore, if you have that adaptation, you are going to be able to survive best in your environment and pass on your genes. And so what we talk about is not survival of the fittest individual or fittest organism, but survival of the fittest genes. The genes get passed on if they are adaptive or beneficial in your specific environment. Every environment's different. Some environments, you need sun protection. That's why we got melanin. Some environments, you need more vitamin D, so you got to have lighter skin. Some environments, it's cold. Some environments, it's hot. Some environments, you need to be able to run fast. Some environments, you need to be able to camouflage. Everything is different depending on the organism, depending on its environment. So these mutations that occur, they're totally random. The mistakes you make when you are replicating your DNA, the A's, T's, C's, and G's, you're supposed to put A next to a T and a C next to a G and A next to a T and then a C next to an A. Uh-oh, that's a mistake. Those mistakes are the mutations, and they can change the protein that is ultimately being made. And that different protein, maybe it's good, maybe it's bad, could potentially cause cancer, but if it's occurring in the gonads, specifically with those cells when they are making egg and sperm, then you might have a zygote that has an original mutation. And then that zygote going through lots of mitosis means every single cell in that developing embryo is going to have that mutation. And then that offspring may have an adaptation, it may not. But this ultimately leads to evolution where nature is selecting the best organism for that environment. Okay? Think we wrapped it. Oh, one more thing. We got to remember that not all cells in your body are constantly making it. I mentioned this before. In your brain, when your brain is working and developing, we don't want new baby cells. We want old, developed, specialized, differentiated, big trees that have grown in your orchard. Okay? We don't want to plant a new cherry tree every year. We want cherry trees to get bigger and produce more and more cherries, okay? That's called G0. They are specializing, they are differentiating, and they are not going through mitosis or meiosis. They are not making new cells. They are working, they are making proteins. That's what we've learned before. When you are making proteins, you are going to be able to do the work of your cell, whether you are a kidney and you need to do work filtering blood, whether you are a stomach and you need to do work making digestive enzymes for your food, or if you are a heart and you need to work to pump, okay? Your muscles, we don't want new muscle cells. We want the muscle cells we have to get stronger, to get better, to get bigger. We want them to have more nuclei, more mitochondria. We want them to have more connections and be better at their job. The working organs and stuff, these are all in G0. And those are not doing your cell division to make new cells. I think I got it all. Thank you.